In agriculture, a soil test is analysis of a soil sample to determine nutrient content, composition, and other characteristics such as pH level. A soil test can determine fertility or the expected growth potential of the soil, which indicates nutrient deficiencies, potential toxicities from excessive fertility, and inhibitions from the presence of non-essential trace minerals. The test is used to mimic the function of roots to assimilate minerals. Labs such as Sugar Research Institute of Fiji recommends that a soil test contain 20 to 25 sample points for every 1 acre of field. As soil nutrients vary with depth and soil components change with time, the depth and timing of a sample may also affect the results. Soil sampling should reflect tillage, past fertilizer or soil amendment placement, cropping patterns and corresponding irrigation requirements, soil type including drainage and slope characteristics, and perhaps old field boundaries such as altered stream beds. When sampling, the major questions to ask are why, when, what, and how. Long periods of monocropping has reduced the nutrient level. Thus, we do soil sampling to determine the nutrient contents of the soil. The most commonly used method of soil sampling would be based on soil types. Fields are split into sampling areas that contain similar soils due to field variations. Hillsides are kept separate from flatlands since the soil types will vary. Also, fields are sampled to know the right amount of fertilizer that needs to be applied. The most appropriate time to sample is before planting the cane, after first plow and first harrowing, or when the farm is in fallow conditions. So what all do you need before you start? A clean spade, preferably marked at 20 cm height, a clean plastic bucket to mix the many borings, a plastic bag to pack the composite sample, soil sampling card and tags to properly label the sample and place in field information. Soil sampling events should be consistent as much as possible. A reference map should be created to record the location and quantity of field samples in order to properly interpret test results. Once a sampling area is determined, remove weeds and vegetation present. Dig a hole to a depth of 20 centimeters. Take samples from the edges of the hole. A sufficient number of cores should be taken to acquire a true representative sample on which you want to plant the cane. This is generally 20 cores per acre using either a zigzag pattern or a grid pattern. Composite sampling can be performed by combining soil from several locations prior to analysis. This is a common procedure but should be used judiciously to avoid skewing results. Collect samples in a plastic bucket, mix it well and sub-sample to 100 grams. Your sample is now ready to be sent to the Sugar Research Institute of Fiji laboratory for analysis. Remember, the quality of the result depends on the type of soil sample you send, so be smart, act accordingly.